Regular physical activity is one of the most consistently validated interventions for extending health span, the period of life spent in good health, and potentially increasing lifespan. Far beyond simply keeping the body fit, exercise operates at the cellular and molecular levels to delay many of the key hallmarks of aging. Aerobic and resistance training both trigger profound adaptations that preserve mitochondrial integrity, reduce systemic inflammation and oxidative stress, maintain cardiovascular and muscular function, and modulate gene expression in ways that mimic some of the biological advantages seen in long-lived individuals. In this context, movement becomes a form of biological signaling, an essential input that shapes how we age, not just how we live. One of the central cellular effects of regular aerobic exercise is the stimulation of mitochondrial biogenesis, the process by which cells produce new mitochondria. Mitochondria are often referred to as the powerhouses of the cell, but their role extends beyond energy production to include regulation of apoptosis, calcium signaling, and reactive oxygen species, ROS, balance. With aging, mitochondria become less efficient and more prone to generating damaging ROS. Aerobic training, however, activates key transcriptional coactivators such as PGC-1A, peroxisome proliferator activated receptor gamma coactivator 1-alpha, which initiate mitochondrial replication and enhance mitochondrial enzyme capacity. This results in greater energy efficiency, reduced oxidative damage, and improved metabolic flexibility, traits commonly seen in individuals who reach advanced ages with preserved physical function. Resistance training, while often associated with strength and hypertrophy, also confers critical anti-aging benefits at the molecular level. It improves insulin sensitivity, enhances glucose uptake via GLUT4 transporters, and reduces sarcopenia, the age-related loss of muscle mass and strength that predicts frailty and mortality in older adults. At the cellular level, resistance exercise stimulates satellite cells, promotes muscle protein synthesis through mTOR signaling in a controlled and adaptive manner, and supports neuromuscular connectivity. Importantly, strength training has also been shown to reduce markers of systemic inflammation, including C-reactive protein and TNF-A, contributing to a more balanced immune state in older adults. Cardiovascular health is another major axis through which exercise exerts longevity-promoting effects. Regular aerobic activity improves endothelial function by increasing nitric oxide availability, reducing arterial stiffness, and promoting angiogenesis, the formation of new capillaries. These adaptations translate to better blood pressure control, more efficient oxygen delivery to tissues, and a lower risk of atherosclerosis and cardiovascular events, still the leading causes of death worldwide. Exercise also favorably influences lipid profiles, increasing HDL cholesterol and reducing triglycerides, while supporting cardiac output and heart rate variability, both of which are strong indicators of autonomic and cardiovascular resilience in aging. At the level of systemic inflammation and oxidative stress, two core drivers of aging, exercise serves as a powerful modulator. Though physical exertion produces transient increases in ROS and inflammatory cytokines, these are quickly resolved and followed by a compensatory upregulation of antioxidant defenses and anti-inflammatory mediators. This hormetic response, whereby low doses of stress induce long-term resilience, leads to enhanced activity of enzymes like superoxide dismutase, SOD, catalase, and glutathione peroxidase. These antioxidant systems mitigate DNA and protein damage and reduce cellular senescence, preserving tissue function over time. Emerging research also shows that physical activity positively affects telomere dynamics. Telomeres, protective caps at the ends of chromosomes, shorten with each cell division, and their length is often used as a marker of biological age. Numerous cross-sectional and longitudinal studies have found that individuals who engage in regular physical activity, especially endurance exercise, have longer telomeres than sedentary peers. This may be mediated by reduced oxidative stress, lower inflammation, and increased expression of telomerase, the enzyme that helps maintain telomere length. Additionally, exercise influences epigenetic regulation. Physical activity has been shown to alter DNA methylation patterns and histone modifications in skeletal muscle, adipose tissue, and even brain cells. These epigenetic changes can upregulate genes involved in energy metabolism, stress resistance, and DNA repair, while downregulating those linked to inflammation and cellular aging. Thus, exercise doesn't just affect current physiology, it rewrites parts of the genetic code's expression, shaping how cells function and respond to their environment over time. The benefits of movement also extend to the brain, which is increasingly recognized as a key organ in aging. Aerobic and resistance training both stimulate the release of neurotrophic factors, including brain-derived neurotrophic factor, BDNF, which supports neurogenesis, synaptic plasticity, and cognitive function. Physical activity enhances cerebral blood flow, improves mood via endorphin and endocannabinoid release, and reduces the risk of neurodegenerative diseases such as Alzheimer's and Parkinson's. These effects are particularly important as lifespan increases. Preserving brain health is now seen as integral to meaningful longevity. Importantly, exercise acts as a unifying modulator of multiple biological systems that tend to decline in parallel with age. Its effects are not isolated to one organ or cell type but span metabolic, cardiovascular, immune, musculoskeletal, and neurological domains. This systemic action reflects the evolutionary role of movement as a core requirement for homeostasis and adaptation. For older adults, regular physical activity, especially when combining aerobic, strength, flexibility, and balance training, helps maintain functional independence, reduce fall risk, support mental well-being, and delay the onset of chronic diseases. In practical terms, the prescription of exercise for aging populations should reflect both biological complexity and individual variability. While moderate to vigorous aerobic activity for 150 to 300 minutes per week remains a widely endorsed target, even modest increases in daily movement can yield measurable health benefits, particularly for those previously sedentary. Resistance training 2 to 3 times per week is now strongly recommended for older adults, not only for muscle maintenance, but also for its systemic anti-aging effects. Ultimately, the science of exercise and aging points to a central conclusion. The body is designed to move, and movement is one of the most powerful, accessible tools for extending health span. Far from being optional, regular physical activity is a biological necessity, one that maintains cellular integrity, supports adaptive capacity, and protects against the many forces that drive age-related decline. In the pursuit of longevity, exercise is not merely supportive, it is foundational.